To begin, I'm going to start by telling you a little bit about what leptospirosis is. Leptospirosis is a zoonotic disease that causes fatal illness in both humans and in animals. Because this one is a little bit more specific, this zoonotic disease can transfer simultaneously between people and animals, meaning another human can also give it to another human, but also a human can give it to an animal, and vice versa. While only around 1 million people annually worldwide are affected, about 58,000 end in fatality. Now, leptospirosis is caused by the spirochete bacterial organism called Leptospira, which comes from the Greek words lepto, meaning fine, and spira, meaning coil. While our project is linked to an ongoing effort sampling female culled cattle for the presence of Leptospira, we decided, because we saw positives in the uterus and urine, can it also be positive in the follicular fluid? In our original project, we found 8 out of 47 positive in the uterus. In this one, focused on follicular fluid, we saw only 1 out of 38, which is 2.6% positive in the follicular fluid. Now, what's interesting about this is the follicular fluid was positive, but the uterus and the urine were both negative. Now, how we confirmed this was by using qPCR. We used the QuantStudio 3 qPCR to detect for the Leptospira in the DNA. Now, how we did this is we took about 0.5 to 1 ml total follicular fluid from each ovary of the dominant or antral follicle. This is the one that is being used to ovulate. After we had this fluid, we spun it down in a centrifuge to get the supernatant and also get any tissue on the bottom. After that, we used the DNA extraction Roche kit to get the DNA. In this, we spun it down about 4 different times, used 2 different washes, and an elution buffer. At the very end, the solution we had left was DNA. With this qPCR, we used Lipo32 and 16S to detect for the Leptospira. We used 2 different ones because they are a little bit different. One is a little bit more sensitive than the other, meaning the heat inside of the qPCR can sometimes cause some upset, which is why we used 2 different kinds to make sure we knew it was positive. Now, on the poster, you'll notice we have a chart at the very bottom. This is the amplification chart. In other words, this is what shows up after the qPCR is finished running. You'll see a bunch of cycle numbers, and this is just how many times it's ran through its heat cycle in fluorescent. Now, the very top 2 lines are 16S positive control. The next higher 2, or the orange ones, are the Lipo32 positive controls. You can see that they have a giant great number compared to the rest. This is because they're positive. They're known to be positive. Now, the one right under the Lipo32 is our sample 32 of a 16S. This one amplified a little bit more than the Lipo32 did, but because both of them had a tiny increase, we can note that they are positive. To confirm this positive, we used a gel electrophoresis to confirm it's positive. Now, you may ask yourself, why does this matter? Well, ironically, leptospirosis lives pretty much everywhere except for Antarctica, but it especially lives here in Tennessee. Right here in Knoxville, it actually lives in the river and all throughout. Now, this is very important for farmers to know because they have to be very cautious about getting an infected cattle. If one cattle gets infected, they pose the risk of the rest of the herd being infected and causing a decrease in their income. Now, while our sample size was a little bit lower and we only sampled at 1 abattoir or slaughterhouse, we continue of this research study. Now, you may ask yourself, leptospirosis, okay, who cares? Well, this actually hits all 3 pillars of One Health. It impacts humans because we're infected. It impacts animals because they're infected. But it also infects the environment. Now, really, it doesn't harm the environment, but because it's in the environment, it causes opposed risk to the rest of the surrounding. It can be in the soil or in the water streams, water basins, etc. Now, if a human gets infected on their farm, and then the cattle ends up getting infected, it causes an economic burden. Now, this economic burden the farmer ends up losing income, but it really has to get rid of the rest of the cattle in case they're positive. Now, there are 4 different types of symptoms that you'll see. You can see this on the left side. There are 4 different types/stages depending on your infection level. While most rodents and farm animals can be asymptomatic, the ones that are sometimes a host, that are the symptomatic ones, they'll have 4 different stages. Now, in your first stage, or your first week of infection, they're asymptomatic, meaning there's really no symptoms showing. Now, in your second week, that's when you'll start to see the fever, the lethargy, the vomiting, the diarrhea. The lethargy comes from the exhaustion of the immune system trying to fight back the disease. That's what a fever is. A fever is an immune response from the disease. Now, thankfully, leptospirosis is very picky. It needs a specific heat, a specific pH, and specific environments to live. Now, this fever can help cause it to die off faster. Now, in your third week of infection, or your stage 3 symptoms, you'll start to see jaundice, pulmonary hemorrhage, renal damage, and hepatic damage. Now, this is when it starts to get a little bit more serious. Now, once you get to your 4th week of infection, this is when you start to see organ failure, or even multi-organ failure. At this point, most of the patients are either too far along to survive, or they just end up dying without knowing it. Now, most people will end up treating this disease with a classic steroid and a classic antibiotic. In vet med, doxycycline is usually the one that we'll try and use. Now, the issue when it comes to people getting infected is most human med doctors don't look for this disease. It's not really that known. Because it looks like a basic any other disease, any other kind of infection, like the flu, strep, stuff like that, that's why it goes undiagnosed for a long time, until usually the 3rd week. Now in vet med, we're more likely to see this, so that's why most people know what it is.